Hey guys, welcome to AP Biology. We're going to do a very quick lecture on cells. So let's go ahead and get started. First of all, um, oops, I want to talk about that we oftentimes need tools in order to see cells because, again, they're really, really, really small. And one of the items that we will be using this next week will be a light microscope. Okay, now light microscopes can only go down to about 1 micrometer, which is 1 millionth of a meter. Okay, and as you can see on the diagram over to here, we can just barely see some of the cell organelles, and below that, we're going to have to use stronger microscopes. Okay, those are called transmission electron microscopes or scanning electron microscopes, and they don't use wavelengths of light. They use electron beams. Okay, so I just want to just show you that those images are much more detailed. However, they're very expensive microscopes, and tens of thousands of dollars, hundreds of thousands of dollars for some of these and things of that nature. But they do make beautiful images that we can use sometimes, and we'll look at some of those a little bit later. All right. So you might wonder, how did we learn about all these various organelles and things like that? Well, we did it by reduction. What does that mean? Is that we reduced the cell below its living sort of level. We isolated the organelles, and we did that by this process we call cell fractionation, which uses centrifuges and blenders and things of that nature to separate out the organelles, and then we can isolate those organelles and we can do studies on them. So that's how that's basically done. And I don't want to spend much more time than that going on it. Okay. Cell theory is important. You need to know what it is. And basically, as I say up here, all organisms are comprised of one or more cells. Now, eukaryotic organisms can also be single-celled organisms. So be aware of that. And some prokaryotic organisms can be associations of multiple cells, not really a single cell, but associations, and they're not really a single organism. We call those biofilms. Plaque on your teeth is an example of a biofilm composed of many bacteria. Cells are the smallest unit of life on the planet. At least so far that's what we know. We may discover something in the future, but right now they are the smallest unit of life. Below the cell level is the organelle, and those by themselves are not considered alive. So once again, you need all those organelles working together in order to have a functioning cell. All right. The other part of the cell theory is that all cells came from pre-existing cells, except the very first cells when the Earth's environment was a lot different than it is today. Some of the earliest cells we know go back to at least 3.5 billion years ago. And we find these in something called a stromatolite, which are basically created in hypersaline environments, super salty environments, where basically these microorganisms, which are prokaryotes, can photosynthesize, or another name for them is blue-green algae. They photosynthesize, and then when the tide changes, they form a thin layer over this rock, and then as the tide comes in, it brings sand and it covers them up, and then the cells grow on top of that, and they form these banded layers. There's some other pictures of stromatolites in Shark Bay, Australia, but this is a fossilized stromatolite at 3.5 billion years old that was found in Australia also. So we know that photosynthesis and prokaryotic cells existed 3.5 billion years ago. Our planet is just under 5 billion years old. So the process of photosynthesis developed pretty early. And again, we only find these things in very unique habitats now, these super saline environments where no other life forms really exist. But again, good evidence of the first life forms. So basically, we have two types of cells. We have the prokaryotic cells and the eukaryotic cells. As you see in the background, we have a eukaryotic cell in the background compared to a prokaryotic bacterium cell up here. And eukaryotic cells are much larger. and then a prokaryotic cell, and then we have a virus, which is a non-living structure or non-living infectious particle compared to a bacterium. As you can see, the viruses are even smaller. But a couple of things right there, just again, eukaryotic cells are much bigger. Eukaryotic cells have nuclei. They can be animal cells, they can be plant cells, they can be fungal cells. Okay, and they can be multicellular or they could be single cellular organisms. Bacterium, again, generally always single cellular, but they can form associations to make these large multicellular colonies, which are visible to us with our naked eye. We're going to get into limits on cell size and surface area to volume ratios tomorrow during lab, and then you also have a MyAP video, so I'm not going to spend much time on that. The only thing I do want you to know right now at this point is that we need lots of surface area per unit volume in order for a cell to work well. Okay. Typical eukaryotic cell, there's lots of different organelles in there. This is a model. It's not what you would really see in real life. Most cells don't have all of these structures, or they might have lots more mitochondria depending upon what the cell structure is. They might not have as much smooth endoplasmic reticulum, so on and so on. So all cells are specialized in their structure. We as humans have over 200 different cell types in our bodies. Each one of them are unique in terms of their structure and in their function. And of course, we can see in a plant also, we see some of the structures there. And we see down here on the lower right hand corner, not in plant cells, things such as lysosomes, centrioles, flagella in mature plant cells. And the same thing in animal cells. We do not see chloroplast, a central vacuole cell wall, nor plasmodesmata. Okay. So you might wonder, how did some of these organelles evolve? How did they come about? How did the eukaryotic cell with all the complex organelles come about? That is called the endosymbiotic hypothesis. As I said earlier, we know that there were cells at least 3.5 billion years ago. Those cells were prokaryotic, meaning they did not have a nucleus, they did not have mitochondria, they did not have chloroplast, they had no membrane-bound organelles whatsoever. So, how did we get those organelles into what's the modern eukaryotic cell? Well, again, the endosymbiotic. Endo means within, sim means together, and bio means living. So living together inside, basically, is what it means. And what we do know is that probably somewhere between 2 to 2.5 billion years ago, proto-eukaryotic cells engulfed free-living prokaryotic cells that either were photosynthetic, such as those blue-green algae, or produced ATP through the process of ingesting other things, bringing other particles in. Those would eventually become the mitochondria, and some would become the chloroplast. Green algae-like structures would become the chloroplast, and the heterotrophic structures would become the mitochondria. So here's a diagram of that. We have two independent bacteria. One bacterium engulfs the other, and now one lives inside the other one. They both benefited from that arrangement, and that made this individual more likely to survive and reproduce and produce more offspring. And we will get into this in more detail, but I wanted you to see very quickly what the endosymbiotic hypothesis is. Other organelles, such as the Golgi apparatus and the endoplasmic reticulum, probably evolved through the process of invagination, this infolding of the plasma membrane, which helps to increase surface area within the cell. And on those membranes within the cell, there are all sorts of proteins and structures and things like that. That's where all of the action happens. So again, here's another picture of invagination. We have some membrane-bound ribosomes. We see the membrane starts to invaginate, to infold, and then part of it encircles the DNA, and part of it will eventually become the endoplasmic reticulum. And that is an example of how we believed some of the organelles have evolved. And again, we're going to go into a deep dive on the plasma membrane this week. And just remember, it is a phospholipid bilayer with the hydrophilic heads facing towards the outside of the cell's environment and also towards the inside of the cell's environment, whereas the fatty acid tails, which are hydrophobic, face towards each other and exclude any type of water. So here's that phospholipid bilayer you can see over here. This is a picture of it, a cartoon. Each one of these little circles with the two long squiggly lines, that is a cartoon figure for a phospholipid. Okay. So again, prokaryotic and eukaryotic cells differ in both size and complexity, but they are similar in these 4 ways. You must know these 4 ways. They are both surrounded by a plasma membrane. They both contain a cytosol, i.e. cytoplasm, which contains all, it's a watery environment which has things like ribosomes in it and stuff of that nature. They all contain DNA, okay, in the form of chromosomes. All right. And they also all have ribosomes. So there's our 4 things: plasma membrane, cytoplasm, DNA in the form of chromosomes, and ribosomes. You need to know those 4 things. Okay. The difference between them is between prokaryotic and eukaryotic cells is the location of the chromosomes. Of course, in a eukaryotic cell, that DNA is contained within a membrane-bound nucleus. In a prokaryotic cell, the DNA is basically concentrated in one area within the cell called the nucleoid region, but it does not have a membrane around it. So make sure you understand that. No membrane-bound DNA in a prokaryotic cell. Again, bacteria are simple, okay? And we can divide the bacteria into 2 groups, gram-positive and gram-negative. In gram-positive cells, and similar to gram-negative cells, they both have, of course, a cell membrane. And in gram-positive, that cell membrane is surrounded by this thick peptidoglycan layer, which is a protein sugar layer. Okay, where in the gram-negative cells, we have a thin peptidoglycan layer, very, 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 very thin, and then it has an additional membrane around it. We call them gram-negative because they don't take up this stain, which is called the gram stain. Okay, and the gram stain dyes the peptidoglycan layer this nice purple color. If the bacterial cells are not purple when they've been subjected to gram stains, then they are the gram-negative ones. And that has, generally, the gram-negative ones can be more dangerous, harder to cure with antibiotics and things of that nature. Okay? And their gram-negative cells are more pathogenic due to the slime layer versus our gram-positive ones. Anthrax is a gram-positive one. Okay, again, what do we know about bacteria? They are very simple cells, no membrane-bound organelles, but they do have ribosomes, but ribosomes do not have a membrane around them. So in bacteria, there's no Golgi apparatus, there's no endoplasmic reticulum, there's no lysosomes, all that kind of stuff. They do have ribosomes, and if they do have a flagella, it doesn't wave back and forth like a sperm cell. Instead, it rotates more like a helicopter blade. Okay. Internal membranes, characteristic of eukaryotic cells. And here's the big important part. It partitions the cell into compartments. Each one of those compartments can have a different internal environment. And those different internal environments allow for specific metabolic functions to occur within a eukaryotic cell. A prokaryotic cell is like a one-room schoolhouse. There's no division within that cell. Makes it very difficult to have a lot of different activities occurring inside of a prokaryotic cell versus a eukaryotic cell. And that is it, guys. I hope this helped.